I would like to introduce our first keynote speaker, Dr. Tal Zaks, the Chief Medical Officer of Moderna. As the Chief Medical Officer, Tal Zaks oversees the clinical development and regulatory affairs across Moderna, the company that's pioneered one of the transformative COVID vaccines that was approved 2 months ago. Dr. Zaks began at GSK in the genetics research group, and prior to joining Moderna, he was the senior VP and the head of global oncology at Sanofi. With Moderna's incredible accomplishments this past year, Dr. Zaks has shown that not all heroes wear capes, some wear coats. And with that, I will turn it over to you, Dr. Zaks.
Thank you, Daniel, and good day, everybody. It's really good to be with you today. I'm going to take the next 20 minutes or so to briefly share the story. Now, I happen to be a medical oncologist. I sort of stumbled into this about 6 years ago. I was looking for my next cool gig, if you will. And I'm somebody who spent a lot of years in training, MD, Ph.D., 2 internships, a fellowship, a residency. I mean, you do the math by the time I was finally to go do something. And what I ended up doing in industry in my career is sort of translating science into medicine. And that's always been my passion, kind of living between the two. About 6 years ago, I saw Moderna and the potential that you could use mRNA as a completely new class of medicine was just something that I couldn't resist, even though it made no sense to my wife. Why would a medical oncologist, given where tumor immunology is, go do this? And the answer is that the era of genomic medicine, if you will, and sequencing and digital writ large has really put us at a very interesting crossroads to translate one of the seminal observations going back 50 years ago, the central dogma of biology, which is that DNA translates to protein through an intermediary called messenger RNA. And put simply, just for the non-scientist in the audience, every cell in our body almost has the same DNA, the same genes, but cells are different, right? My skin cell and my eye cell and my bone cells are different. And why is that? Well, because different cells make different proteins. But if all cells have the same DNA, how do they make different proteins? And it turns out that every cell at any given time makes only a fraction of what they know how to make, and that fraction is determined by the message that encodes the right information from the DNA to the cell's machinery, the ribosome, to go make protein. So if you could actually make a drug or a vaccine that is based on information that comes in the middle of that process and basically tells the cell, go make this protein, and you design a protein with a therapeutic application, you opened up an entire new class of medicines. And that opportunity is really what set Moderna going. And as you can intuit, what it would mean if it would work is that we would have basically a sequence-based or an information molecule as a drug. And so all of our messenger RNA drugs and vaccines all are fundamentally the same thing. They're a molecule that has a certain code, and that code determines what protein the cell will make. Change the code, you get a different protein. So this now allows us to think of a whole world of applications that start with the cell making their own protein based on the instructions that they've been given. Now, if you step back and ask yourself, okay, what can I do with this? You quickly realize that actually vaccines is one of the low-hanging fruits for this technology. Now, stepping back, we've done vaccines, we're doing rare disease, we've done things in cancer, our collaborators are doing things in the heart. So the world of application is very broad, but from a very simplistic point of view, vaccines are sort of the easy thing to do. And the reason is twofold. Number one, for viruses, we sort of know what protein we're trying to target or what is it that we want the cell to make, because it's pretty straightforward to sequence a virus. And we know from the history of vaccines across many different types of viruses, what is the protein that should serve to teach the immune system to recognize the virus, what's called an antigen. And the second reason is that you actually need only a smidgen of protein made, and the delivery system is 100 years old or more. It's basically a syringe, which you inject into the muscle. The mRNA goes there and teaches the cells, our own body cells, to make the protein. In this case, it's the protein of a virus. And so we've been at this now for several years, turning this platform into a potential vaccine platform. And indeed, we started back in 2015 with an influenza, and over the years have shown time and again in early small studies that we could indeed teach the immune system how to recognize the antigen, because the immune system time and again would generate antibodies that had neutralizing activity against the virus, whether it was influenza, a human metapneumovirus, parainfluenza virus. There's a whole bunch of different viruses that we've shown in early phase 1 trials. And so the potential for this platform then to serve as a platform for pandemic preparedness wasn't lost. And it's not a mistake that our first candidate in the clinic back in 2015 was against influenza. It was a pandemic threat of influenza. And so we've been working in collaboration with the National Institutes of Health over the years. And as luck would have it, at the end of 2019, September of 2019, the CEO, Stefan Bancel, and I were down meeting with Tony Fauci and his team, reviewing our clinical data. We had been collaborating with the NIH on the cousins of SARS-CoV-2, which at the time were SARS-CoV-1 and MERS. And we had shown in animal models that indeed this platform would work. We'd shown in the clinic, as I said, in the 8 viruses that we had targeted to date, in all 8 against which we were successful generating neutralizing antibodies. So we had a pretty good track record. And at the time, at the end of 19, the discussion with the NIH was, let's do a demonstration project and see if we just artificially start the clock, how fast can you guys go should there be a pandemic? Unfortunately, as luck would have it, within a couple of months, it became clear at the end of December, beginning of January, that indeed what we're facing is actually not a drill. This is live fire. And so because this is really essentially an information molecule, within 48 hours of the sequence being known on the web, we had designed and put into production our first vaccine. And here we benefit really from standing on the shoulders of giants before us, in the sense that the scientific community had already understood that it's the spike protein, that's the part of the virus you want to really teach the immune system to recognize. And so it's not a mistake. If you look around you, all the vaccines coming to market these days are essentially using the spike protein as the antigen to teach the immune system to recognize the virus. And so we put that within 48 hours into production. We partnered with the NIH to do the phase 1. As soon as we had data from the phase 1 to inform us of safety, we started the phase 2. As soon as we had data still from phase 1 to inform us of the dose, we collaborated again with the NIH to start a phase 3. And so the phase 1, the data, the sequence data was January 11th. The phase 1 started in March. The phase 3 started towards the end of July. And the phase 3 study was a very large, conservatively run trial. It gave results quickly, and it gave results quickly because it's a case-driven study. And so when you want to prove that a vaccine can actually prevent disease, you need to see enough cases of disease occurring in the people getting placebo, and you compare it to the lack of cases in the people getting vaccine. In our case, we were conducting the trial during a time of high transmission rates in the United States, and so the trial read out, unfortunately, quickly. And I say unfortunately because the reason it read quickly is that we had so many cases occurring. The efficacy that we had demonstrated was remarkable. And here, if you kind of step back, when we as scientists look at trial results or any scientific result, the first thing we ask ourselves, is this a fluke or is this true? And we had an efficacy of 94.1%. Well, we are not alone, and the same idea, the same scientific principle, even the same spike protein antigen by design was developed by a small German company in collaboration with Pfizer, and they had run a study parallel to our own. And what you see here is 2 companies, completely independent by law, we don't collaborate, each having their own technology, and you'll see some of the manufacturing infrastructure lead to differences in cold chain requirements and others. So clearly we have a different product. But we're both using the same scientific principle. We're both trying to immunize people to prevent disease. And both of these trials read out within a week of each other, and the efficacy estimates were within 1%. 95% for us, 95% for them, 94.1% for us. That level of scientific replication, everybody asks me, what was different about your vaccines? And I'm sure we can get into it. But the mind-boggling element is how similar the results were and how high the efficacy is. And I think that's the highest degree of scientific replication I've seen in my career, and certainly the most important one, because it underpins the veracity and expectation of indeed benefit. Now, that was back in November, December. Since then, Pfizer's vaccine has been authorized and approved across the world, as has ours. To date, there have been roughly 27 to 30 million people in the US dosed with each, so a total of well over 50 million people. These vaccines have side effect. Ours will cause you some local pain on injection. After the second injection, we see a little bit more of systemic symptoms, fatigue, and some muscle aches. This is uncomfortable, but it is not dangerous insofar as that it's expected and it's transient and it'll go away. It's a hallmark of effective activation of the immune system. And in fact, only this morning, we saw the first report come out of CDC looking at the first month of safety as these vaccines have been deployed in the United States, and basically saying there's no big surprise here. There's nothing unexpected. Even the cases of anaphylaxis that initially people were concerned about, according to the CDC, seems to be the same rate of anaphylaxis that you see with every other vaccine. And so we're now in the midst of a global launch, really, for this vaccine. There have been, there is continuing a lot of work and a lot of questions. When you get a vaccine approved so quickly, obviously the longevity of the immune response is yet to be demonstrated. And so we continue to follow the participants on our trial. I think what's been reassuring is that we've seen recently data come out from what's called real world evidence. So people looking at populations that have been immunized and then following cases. The best data have thus far come out of the state of Israel in terms of monitoring the actual infection rates and comparing them in their population between those that have been vaccinated and those that have yet to be vaccinated, and already are showing a dramatic impact on the rates of infection. Given that these vaccines, the mRNA vaccines and ours especially, tend to cause or elicit even a higher rate of neutralizing antibody that you see following infection, we anticipate that these vaccines are going to be durable. The immunogenicity we see should be durable, I estimate, for at least a year. But I think that story is now a little bit been put into question with the emergence of some variants of concern, namely those that have emerged in South Africa and Brazil, where you can see that people who've been either previously infected or immunized can still neutralize the virus, but there's some drop in the ability to neutralize these variants. And so there's a question of whether over time, if the immunity wanes, will it wane first against those variants, and will those variants start to circulate? And so there's a lot of questions ahead of us that we need to answer. And we have already said that we are going to put into clinical testing a vaccine based on this variant. Remember, our vaccine platform is essentially just a code-based, it's an information molecule, so changing the code to adapt it to a new variant is pretty straightforward. And since we now have the manufacturing infrastructure, it should be a matter of weeks to be able to just change wholesale the vaccine should we need to. Although at this point, I would say this is for us more as an insurance policy, and potentially, if we need to in the future, we would be able to further boost people with a variant-tailored vaccine. I think as I step back and look at last year, there's been a couple of learnings that I think are going to stay with us that are worth reflecting on. And first and foremost has been the tremendous level of collaboration that we've seen across not just industry, but with all relevant stakeholders. The big unsung heroes of this revolution are actually the FDA. I mean, if you think about it, I led the development of our vaccine, but I was able to sort of triple and quadruple my team as I was building it to make sure that I've got all the resources I need. I could go partner with a contract research organization, PPD, who did a tremendous job and at one point had about 1,000 people on our program. The FDA has had to look at the same data with the same staff, and in fact, at the same time, had to look at data of other competitors, and the rest of their work hasn't stopped either. And so the ability and the richness we have in our country of having such a dedicated civil service, I think, is really admirable. And I think we owe them all a debt of gratitude for how they've stepped up to work with industry and slightly ahead of industry, providing guidance and support along the way. In our case, we've been helped by the US government, by BARDA sponsorship, and we've worked closely in collaboration with, as I said, the National Institutes of Health, not just on the science, but also on some of the less sexy stuff, if you will, the biomarker work. Imagine when we started the first vaccine, if somebody would have been vaccinated, we actually had no assays to tell us whether they're immune to the virus or not. All those assays had to be built from scratch to very rapid timelines. And this is where I think the expertise of academic labs and our NIH labs have really come to support not just us, but the field as a whole. I think this has been true globally as well. There have been other countries that have stepped up, but I think in many ways, in transparency, the US has led the way. And I'll close by just making a couple of points on sort of the conduct of trials in this era. This level of collaboration is also a level of collaboration with the public, I think. As we were launching our phase 3 trial, we put a lot of effort in ensuring that our trial results will be reflective of the population that actually needs the vaccines, which means we want to make sure we have enough older people. We want to make sure we have people with comorbidities, and we want to make sure we have enough people from minorities and underserved population. And as is well known, many of those populations actually don't trust anything coming from pharma or the government. And so it took some time to build that trust. And I think this trust in the data and trust in what we're doing continues to be a key factor as we roll out the vaccine. For us as a sponsor, I think the realization was that one of the most important tools that we have is transparency. So I'm very proud of the fact that we were the first company to actually publish our protocol on the web, unredacted, as it was going. And not only that, but we published the metrics of recruitment on a weekly basis so people could follow and see our progress and see who are the populations we were recruiting every week and watch the trial and evolution. Similarly, there was a great public debate and discourse on the trial results with an external group of expert advisors that the FDA called to, where FDA and us spoke about the results. And I think that work is only sort of beginning as we have an opportunity ahead of us in the sense that no time since I've been an adult, I'd say, has the public been this interested and this involved in science and understanding what this virus means, what do vaccines mean, how does drug development work, et cetera. And I think it's an opportunity for those of us who care about translating science into medicine to make sure we not forget the last step. The medicine isn't going to get there unless you translate the medicine into politics. And politics here, not as a bad word, but politics is really the thing that unites us all and allows humans to find shared meaning in what they do. And in that regard, it's an honor and a privilege to be speaking to all of you here and be able to answer any questions, because I think through transparency and understanding and just discourse is going to be critical for the adoption and understanding of the impact we hope to have on the public's health by changing around the shape of this pandemic with the mass vaccination effort that I think is unheralded in our lifetimes. So with that, let me conclude my soliloquy, and I look forward to the conversation.
Okay, I don't know if Daniel is going to introduce me, but I'm Marvin Lieberman. I'm a professor at UCLA Anderson. And Dr. Zaks, thank you very much for agreeing to be our keynote speaker, and thanks to Daniel for involving me as the moderator. I've been presented with a list of questions that were submitted, and I would have to say that Dr. Zaks has answered a lot of these, but we still have a very good list. Let me begin, as you did, Dr. Zaks, with a kind of a broad perspective on messenger RNA vaccines, on the Moderna platform, on your pipeline. So So one set of questions relates to the success of messenger RNA vaccines. Yours and Pfizer both, as you say, exceeded what people's expectations were. And I imagine that validates much of your broader platform. As you mentioned, you have vaccines and potentially other therapies for many kinds of diseases. So the first question here is, how does the success of the first vaccine you have, contribute to the rest of your platform? And then more broadly, if you could talk about competition among different kinds of vaccines. I think what we've seen in this case, a surprise to many, we've got a horse race of many different approaches to vaccine development. And the first out of the gate were the messenger RNA vaccines. You talked about why it was possible to develop those, so quickly. And they've also turned out to be extraordinarily efficacious. So, it raises the question of, as these other vaccines come to market from other companies, what's likely to happen both in the COVID-19 space in terms of competition, and more broadly, does this suggest that messenger RNA vaccines are the wave of the future for vaccines in general? I think you're muted.
Sorry, famous last words. Those are two big questions, so let me take them one at a time. So I think the success of our vaccine, well, first of all, it's predicated on an anticipation of success that, for those of us close to it, we're not terribly surprised. And I say this humbly because the ability to translate neutralizing antibodies into preventing disease in cases of infectious disease vaccines has been well predicated for many years in many, many different vaccines. So in a way, it was reassuring. But once we saw the phase 1 results, and we saw the very high level of neutralizing antibodies, we had a strong expectation of benefit. Now, that being said, I think there are a number of fronts where this validation is really critical. The first is in the public's eye, right? So if you recall, back in the second half of last year, there was a lot of questions. Well, it's a new platform. We don't know what's going to happen. Well, 60 million people immunized later, and a CDC report that says safety is as expected so far, I think is tremendously reassuring to everybody, the public at large, and certainly regulators. And I think that makes life in the future much easier for the follow-up vaccines. We have a cytomegalo vaccine that is going to enter phase 3 this year. So there's many more vaccines to follow. The second success, or the second impact, if you will, is the manufacturing. And people, most of us focus on the science and the biology, but I think one of the strengths of our company is that our CEO is actually an engineer with a background in manufacturing. Years ago, we had invested heavily. After we saw the results from the first vaccine, we decided to take about, a sixth of our balance sheet at the time and put it in an infrastructure, in a manufacturing plant, because we believed that having control and understanding what are called the critical quality attributes as you manufacture a vaccine would prove critical. And indeed, if you see what we've come to market with, we've got a vaccine that is stable at a temperature we all have in our fridge at home for 6 months. It's stable in 4 to 8 degrees Celsius for a month, and that's all a product of investment in the manufacturing side. And those elements are critical. So we now have the manufacturing footprint for success as well. And the final piece I would say is, it's proven to us, as I look at the pipeline of products, that we have an anticipation of a high success rate in vaccines, but we've also clearly demonstrated in a way that's unimpeachable that when we teach our body how to make a protein based on the sequence, then the body makes the right protein, and that protein has the right pharmacological effect. Now, everybody who's close to it says, yeah, well, of course, the body can't make the wrong protein. Nonetheless, the proof in 30,000 people in preventing COVID-19 disease, I think makes everybody much more comfortable that that's not just a scientific principle, we've actually reduced it to practice. And it's that same principle that should apply in those other applications, whether it's children born with a rare genetic disease because they're missing a gene or they've got a mistake in a gene, and we can actually put in a messenger RNA that will go into their liver and translate into protein and thus correct the disease. Or whether our collaborators at AstraZeneca are using messenger RNA to encode for a growth factor and are injecting it directly into the hearts of people after undergoing coronary artery bypass grafting, so that the repair is much accelerated and there's less congestive heart failure after the procedure. That's an experiment ongoing. So I think the success will clearly increase the perception of the public and the anticipation of more positive results from the rest of the pipeline. Now, you asked me a question about competitors and other vaccine platforms in the future. And I think you're already seeing that in COVID. There's a really unique feature of this vaccine platform that, to be honest, we didn't entirely anticipate, which is there seems to be a benefit in focusing the attention of the immune system just on that antigen, but doing it with a platform that mimics biology. Now, why do I say this? Well, an mRNA translates protein from within the cell. And by doing so, that protein not only gets displayed on the cell surface, but it actually gets chopped up and displayed to a different effector arm of the immune system called T cells. And so it's just like viruses, and it's not a mistake that the most prevalent class of viruses are RNA viruses. So think about what our platform is. We've got a messenger RNA encapsulated in a lipid nanoparticle. And to eons of evolution, that's a virus. The trick that we and others have figured out is how to, once it gets inside the cell doesn't think it's a virus. The cell believes that that's kosher messenger RNA that it can go make protein of. And so once we get under the tripwire of recognition of that messenger RNA as foreign, now we've got a platform that can make proteins, but we're still stimulating both T cells and antibodies. We're still making a very robust immune response. Now, why do I say that? If you look at another application, our CMV vaccine, our vaccination, in CMV is a case where some of us are chronically infected. We walk around with levels of antibodies in our blood every day. It turns out that with an mRNA vaccine, you can do something that people thought was not scientifically achievable. You can supersede the level of immunity that somebody who's chronically infected has. And so if you take a naive person and you immunize them, you not only get to the level of a latent infection, you actually exceed that by about a log. And if you take somebody who's already infected, you increase their level of antibodies. So there's something about the ability of this platform to focus the lens of the immune system that is really powerful. And I think that's what underpins the fact that the success rate, the point estimate for efficacy, the 94, 95% we're seeing, is so far the highest that you've seen for any other technology, whether it's protein or adenovectors. I do think that as I look into the future, people are already, and you're seeing this come out of Europe, there's an emerging sense in the public that there's a difference in the efficacy between the adenovector vaccines, namely AstraZeneca's in this case, and the mRNA vaccines. Novavax has a good protein vaccine. Their point estimate for efficacy is 90%. It's fairly strong. It's anticipated by their neutralizing antibodies. And I think you will see additional protein vaccines, protein subunit vaccines come to the fore. But I think that the future of messenger RNA vaccines certainly looks bright in comparison, both in terms of the speed and in terms of the magnitude of benefit that we see. Is it going to be for every type of antigen? Probably not. I mean, I can think of bacterial molecules that can't be made using messenger RNA. So maybe for those, it'll take us time to figure out what the right antigens are. But certainly in the vaccines against which we're working, we see a very high probability of success, and we see the ability scientifically to differentiate from those other types of vaccines.
Very interesting. In the 10 minutes that we have left, why don't we turn to to more current issues with COVID-19. I think one, it relates to these new variants, which you mentioned, the South African variant and the Brazilian variant, and presumably these variants will continue to appear. And looking to the longer term, I mean, Moderna must have a strategy to deal with that. Will we get boosting doses? Will this become kind of like the flu vaccine, where every year you need to have another shot? And then again, related to that is will COVID ever completely disappear, or are we going to be in this cycle for, potentially decades?
So in my heritage, there's a very well-known saying that since the destruction of the temple, prophecy has been given to fools. So from this point on, you should consider me a fool, but to the degree that you listen to me, well, that's on you. If I look at other coronaviruses, we as a human population have been living with coronaviruses for at least the last 100, 150 years, as far as we can tell. And there's 4 other ones circulating, and they cause respiratory illnesses, mostly in the very young and the very old. Not very frequent, probably together they account for less than 5% of such respiratory illnesses, but it can, in an immunocompromised individual, in an old person or very young, cause significant disease. It turns out that, the herd immunity here is imperfect, and plus we, over time, immunity against them tends to wane and we can get reinfected. And it's unclear to me whether that's a function of having very mild disease and therefore the immune system just shrugs its shoulder and never mounts a very strong response, versus having more severe disease that would be associated with a stronger immune response. Now, the other side of the, the other extremity, the other extreme example is the Spanish flu. So it came in 1918. By 1922, it was gone and hasn't been seen since. We've got other flu variants, but not that one. So I think for us, for coronaviruses, the truth will probably be somewhere in the middle. I think in the coming years, you will see continued threat from SARS-CoV-2, whether it's this variant, it could be in the future additional variants that will emerge. I think it's important to note that the variants that we see now are probably emerging more because they have improved fitness to infecting naive hosts, although there is a sense that the South African strain and the Brazilian strain also have a component of fitness to an immunological response. So there may be some element of an immune escape there. So I anticipate, to make a long story short, we may need to get boosted within a year or two, probably against a strain-specific vaccine. And in the future, there may be evolution of this virus into strains that would require additional boosting over time. Whether it becomes like a flu, I don't know yet. I think the sense is that it's not mutating as fast as flu does, so maybe not quite every year, but I think it will be with us. I think the other challenge is it's truly a global problem. There's no place on the planet that has not seen COVID. And so it's going to take us time to immunize the globe. And as long as we haven't, it will continue to circulate in some corners and poses a threat for us. So that's my sort of personal prophecy here. Now, your question was, what are we doing about it? Well, we have the benefit now of having stood up a manufacturing infrastructure, having a sequence-based platform. So it's very straightforward for us to introduce a variant-matched vaccine. And that's exactly what we're doing, so that we are ready as a company and as society to boost or immunize de novo people who are still naive next year with the best kind of vaccine that we can come up with that's as closely matched as possible to the strains that are threatening us.
Let me turn to the question of capacity, which you emphasized a bit in your talk. Moderna has, as you said, an engineering CEO who's focused on production issues, but clearly in the short term, the capacity constraints are binding. It's really a serious issue. I mean, ultimately, we may have oversupply of vaccines, but in the short term, increasing capacity is really critical. Can you talk a little bit about what Moderna is doing along those lines?
So we continue to work to increase our capacity, and increasing capacity is a function of both investment, but also where the bottlenecks are. Today, there's the bottlenecks alternate between having enough glass vials, having enough plants, manufacturing facilities that can actually finish those vials. Then it'll turn to having enough of the right kind of needles to extract the volume from those vials. So that work continues, and I think we're continuing to expand our manufacturing footprint. I do anticipate with the advent of more protein vaccines down the road that there will be a surplus capacity at some point, and it will become much more commoditized, but that's probably going to happen. We thought initially that would happen, towards the end of this year. I think that that's probably going to be more of a 2022 and beyond luxury if we can get there.
All right, thanks. I don't know if you can comment, but today there was some interesting news from Pfizer. They did a trial in Israel, I believe, that showed that the Pfizer vaccine, which is similar to yours, is highly effective after just a single dose. And what's more, their vaccine, which previously had required really low temperatures, minus 80 degrees centigrade, can actually be stored in regular freezers. And I assume that Moderna must be doing similar tests that, ultimately we'll see some relaxation of the dosages and the freezer temperatures that will, make it much easier to roll out these vaccines around the world. Can you comment on that at all?
Yeah, sure. So let's start with the freezer requirements. They've gone from needing minus 70 to being stable at minus 20 for 2 weeks. Now that's just 2 weeks. The problem is you got a 3-week interval between prime and boost. So that still has an impact on the supply chain. Our vaccine is stable for 6 months at minus 20, and after that for a whole month at 4 to 8 degrees in the fridge. So I still think, while I commend them on the improvement, if you just draw the comparison, you see there still is a lot that can be improved, and I trust they will. Now, in terms of a one-dose efficacy, here's the challenge. What we've seen from Israel is similar to what we had seen on the clinical trial, insofar as that you look at anybody who's been vaccinated for the first dose and you follow them for the next 28 days, you indeed see that first dose is already having an effect, and that's terrific. We've seen that in our vaccine as well, was part of the phase 3 data. We also see a reduction already on asymptomatic infections, not just the symptomatic and the severe. The challenge is that we don't know how durable that's going to be. And here's where the science, I think, is informative. Messenger RNAs are transient. They don't change our bodies. They're just a transient message. Within a day or two, they're gone, and the protein's gone within 2 to 3 days. And so just showing that, while it has a nice effect in the short term, I really believe that you want to come in with a second dose a month later and cement that immune response and make sure that you get the right memory. And so what we don't know from these early data is how durable that is, because everybody then gets the second dose, or practically everybody does. So my recommendation is anybody contemplating vaccination should get the 2 doses from a messenger RNA the way they were designed in the trial. It may be different with an adenovector, because that hangs around in your body for longer after the first dose, and the relative benefit of the boost is less than it is for an mRNA-based vaccine.
Okay, very interesting. We've got just a few minutes left. Let me ask you a closing question about, you talked a little bit about public involvement. There is a real question about communication around the vaccines. I think it's only appropriate to say there's been a huge amount of confusion over the past year about vaccines and other therapies. There's a big difference between what the public can understand and what professionals, the FDA and MDs like yourself and others in the field can understand. Is there an appropriate way that Moderna can be part of that conversation to help to educate the public?
So first of all, I would take exception to your sense that there's a big difference between how FDA or a physician understands this and how the public understands it. I think we underrate the public. I think the public has a very good understanding of it. I think everybody comes to that understanding through their own lens and their own biases. What I found is we really have only I, as the chief medical officer, at the end of the day, have two tools. One is transparency, which I alluded to, which means you're welcome to look at my data anytime, and we've shared them. We've been public about it. We publish them, etc. The second is, to a certain degree, an example. So I can tell you, my mom, who's 80, got the vaccine. She's in Israel. She called me up. She said, "Hey, yours isn't here yet. It's the Pfizer's vaccine. What should I take?" I said, "Mom, you take any vaccine that's being offered. It's certainly better than none, and their vaccine looks as good as ours." Remember what I said about the scientific replication. My daughter-in-law, who's a physician in New York City and has been in the front lines of COVID for the past year, has gotten vaccinated. And so beyond that, it's engagement with the public. The public ultimately trusts the stakeholders that they trust, which tend to be at the local level, politicians at the national level, et cetera. And so we try to work through those stakeholders as a company, whether it's FDA, CDC, trusted officials, all the way down to the local level, leaders that people trust, in order to explain, engage, and sort of make the science accessible and understood to everybody.
All right, we are exactly on time. We have a 10:30 finishing time, but this has been fascinating. I mean, thank you, Dr. Zaks, for a tremendous amount of information in a short period of time. I mean, I think Moderna has been quite remarkable. You know, from a business school, you know, we admire them as a highly innovative and successful business, and one that's literally helping to save the world at this point. And you know, we're looking forward not only to your success, continued success in the COVID-19 vaccine, but this whole platform that's been validated here that can be rolled out, as you indicated, to many other threats to human health. I myself am scheduled to get my first vaccine shot next Wednesday. I don't know if I'll get the Moderna shot or the Pfizer shot. As you said, either one will be great. But I'm certainly looking forward to being on the other side, where I can actually go out of my house, go shop for groceries, whatever, and not worry about contracting COVID. So again, thank you so much. We've got a lot of things to think about here based on your talk, and we've all learned a tremendous amount. So thank you again, and thanks to the organizers for setting all this up.
It's been a real pleasure. Thank you. Thank you for having me. Have a good day.